Hi, my name is Lauren Van Oudenberg, and I'm presenting on a newly discovered genetic abnormality that could possibly be used in the development of a male contraceptive. Dr. Richard Smith of the University of Iowa published these findings in the April 2nd edition of the American Journal of Human Genetics. Female oral contraceptives have been developed for over 40 years and have become extremely commonplace in today's society. However, no similar pharmacological contraceptive has been developed for males. Caspr-1 is a gene that involves non-syndromic male infertility in humans, which means that the infertility is not part of a syndrome. This could lead to future therapies that replace the gene or the protein. The research team collaborated with scientists from the University of Social Welfare and Rehabilitation Sciences in Tehran. They discovered the male infertility gene while studying the genetics of families from Iran. Iranians have a relatively high rate of disease-causing gene mutations seen within their population, which makes them very interesting to study. In this particular study, they noted two families where male infertility was not a part of a syndrome and appeared to be inherited. From there, the researchers focused on a group of genes that have been implicated in male infertility in mice, and were able to find that the mutations in both Iranian families occurred in a single gene called Caspr-1. Harvard University did further studies on mice that lack the Caspr-1 gene to reveal how sperm is affected when the protein is missing or abnormal. These studies found that the Caspr-1 mutations affect sperm motility, or the ability to move spontaneously and actively consuming energy in the process. Specifically, they noticed that the very vigorous hyperactive motions that sperm use when entering an egg during fertilization was compromised. Research suggests that the defect in sperm hyperactivity that's seen in mice without the Caspr-1 gene will also occur in humans with the genetic mutation. There are several studies looking into possible male contraception, including one that uses the Caspr-1 gene. The Caspr-1 approach is what's called an immunocontraceptive. An immunocontraceptive is a birth control method that uses the body's immune response to prevent pregnancy. One of its most popular uses is to control populations of wild animals. It's also designed to provide long-term, not permanent protection. One of the biggest benefits of an immunocontraceptive is that it has a lack of an endocrine or metabolic side effects, which makes it easier for people to take, and it doesn't require insertion or an implantation of a device. The way an immunocontraceptive works is that it develops antibodies that bind to a target protein and block its function. Antibodies are proteins that bind very tightly to their antigens. They are produced as a defense against infection. When foreign materials enter the body, specialized cells in the lymph system recognize it as alien. The recognition of the foreign material or antigens triggers a chain reaction of immune responses. Cells in the lymph system, known as type B lymphocytes, produce an antibody and made to match that particular antigen. For the immune system to recognize the cell from its own body as foreign, a foreign substance must bind to the targeted cell. This tricks the body into thinking that its own cell is an antigen. The duration of the contraceptive would depend on how long the antigen can remain bound to the antibodies. Essentially, immunocontraceptives require the production of a controlled, time-limited, and non-pathogenic immune response to components of the reproductive process. Immunocontraceptive development is still in its very early stages and needs to be proven effective, safe, and reversible before it can be marketed. However, the fact that the Caspr-1 gene has been identified as negatively affecting sperm motility means scientists are well on their way to discovering how it could be used as a male immunocontraceptive.